So I just summed up glycolysis over the course of stage 1 and stage 2, phase 1, phase 2. Or stage 1, we took glucose, which is indicated right here at the top, all the way down to 2 molecules of glyceraldehyde 3-phosphate, which are both displayed right here. Now what we're going to do is we're going to look at each one of the enzymes that is involved in those processes. So our first enzyme is one known as hexokinase. And what hexokinase does is it takes glucose along with ATP and it produces glucose 6-phosphate. You'll see that displayed here. Step 1 in stage or phase 1 of this entire process is this enzyme right here, hexokinase, which takes this molecule right here, glucose, and it is going to do a nucleophilic transfer reaction where we take ATP and we transfer one phosphate molecule onto carbon number 6 of glucose, and we're going to produce an aptly named glucose 6-phosphate. Now this is a metabolically irreversible regulated step. This is the first investment in ATP. I'll use that terminology a little bit, investment and payback sort of thing, because we've looked at the overall net of this process, but this is the first place where we actually have to spend in ATP, spend money to make money, spend ATP to make money, ATP, sorry. Okay, so our next step, after we've taken glucose, our 6-carbon sugar, and we've made glucose 6-phosphate, we're going to use this enzyme known as glucose phosphate isomerase, or you'll sometimes see this labeled as GPI. Now, one thing I'd like you to notice about this is that this is an isomerase, so we're simply rearranging all the positioning of our atoms. This is an irreversible step, this is a reversible step. So we have this kind of we have this equilibrium arrow that shows that this can go basically backwards or forwards. Our conversion of glucose 6-phosphate into fructose 6-phosphate. We've taken it from a 6-atom ring and we made a 5-atom ring. So basically what that means is we took carbon number 1 and we put it out here by itself. Previously in glucose 6-phosphate, carbon number 1 was a chiral carbon. This was our anomeric carbon. Now what we've done is we reposition this, so carbon number 1 is outside the ring. It is now achiral because it has 2 hydrogens to which it's directly bound. And what this means is now we've got a little bit of symmetry almost. If you look at carbon number 6 versus carbon number 1, they're kind of similar to one another. So that's our staging for our next step, which is catalyzed by the enzyme phosphofructokinase. Again, taking ATP, investing in ATP to phosphorylate fructose 6-phosphate to make fructose 1,6 bisphosphate. So here is our F6P, fructose 6-phosphate, and here is the resulting molecule F16BP, fructose 1,6 bisphosphate. We've created something that looks pretty much symmetrical. So phosphofructokinase is the enzyme that does that. It invests our second ATP molecule. This is what is known as the first committed step in glycolysis. So what that means is, one, it's irreversible, but also whenever you make this fructose 1,6 bisphosphate, that molecule is going to be going on through glycolysis. If you compare it to step number 1, our hexokinase, that was an irreversible step, but that fructose 6-phosphate could go in any number of different directions. Whereas with this reaction, this is saying fructose 1,6 bisphosphate, this is kind of an extreme, but it only has one purpose, and that is to go through glycolysis. Glucose 6-phosphate, on the other hand, could go in a number of different directions. So that's what we're looking at here. So what we've got here is a total of 3 steps where we've taken glucose, we've phosphorylated it twice, and we've done an isomerization. Not in that exact sequence, but this is where we are. Okay. Now our next step, where we've created our fructose 1,6 bisphosphate, that's almost a symmetrical molecule. Now we're going to use an enzyme known as aldolase. And what aldolase is going to do is it's going to take that symmetrical 6-carbon molecule and boom, break it into 2 3-carbon molecules. So, and I use symmetrical kind of loosely. This is not a perfectly symmetrical molecule, but I want you to see a carbon 1, 2, there, 1, 2, there. Each carbon, each one of these kind of achiral carbons has a phosphate group on it. The others are chiral. Okay, so aldolase is an enzyme that's going to do an aldol cleavage reaction where we're going to break this into two different molecules. We're going to make one molecule known as dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is kind of the molecule that we talked about as the end of stage 1. So how do we get there? Well, it's kind of simple. These two products, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, kind of have two different roles. Once glyceraldehyde 3-phosphate is produced, it's ready to go. So 3 of the carbons resulting from our fructose 1,6-bisphosphate are ready to go on to be metabolized further. They have reached the finish line of stage 1. Dihydroxyacetone phosphate, on the other hand, well, that has to undergo an isomerization. Triosephosphate isomerase is going to take that molecule and rearrange everything on there. It's going to take it from dihydroxyacetone phosphate to make another molecule of glyceraldehyde 3-phosphate. We'll see that displayed right here. Here is our dihydroxyacetone phosphate. We've got 3 carbons. What we're going to do is we're going to make an ene-diol intermediate, and then we're going to make glyceraldehyde 3-phosphate. So essentially, what this consists of is taking this double bonded oxygen and shifting it up. Taking this hydroxide group that was on that carbon and shifting it down. Now, what this is going to yield is the end of stage 1. We have completed stage 1, or phase 1 of glycolysis. It took us a couple of different reactions. It took phosphorylation, isomerization, phosphorylation, a split, and then an isomerization. So if you think about stage 1, think phosphorylation. You got to rearrange them, so you use an isomerase. You got to phosphorylate. Then, you got to do a second phosphorylation. Then you can break it into not completely symmetrical identical molecules, but very similar molecules. Once you've broken those into 2 3-carbon molecules, you can make those 3-carbon molecules identical. And that's where triosephosphate isomerase. So phosphorylate, rearrange, phosphorylate, cut, rearrange. That's it. Those are the steps to get through stage 1 of glycolysis. All right, I'll see you in the next stage.